Introduction, tendon progenitor cells for rotator cuff repair. Now, pre-injury, the rotator cuff tendon-to-bone attachment consists of 4 distinct layers, and it's a transitional gradient between those layers that facilitates the load transfer that arises from the muscles of the rotator cuff. Post-repair, the distinct structure organization is lost, and the healing tissue is characterized by fibrovascular scar tissue with inferior biomechanical properties, which predisposes to re-tears. Now, the fundamental limitation of rotator cuff repair is not being able to recreate or regenerate the native enthesis. And the question is, how can we make a shift from pro-fibrotic to pro-regenerative healing happen? Now, previous research from the Mount Sinai lab has shown in a neonatal Achilles tendon transection model that neonatal mice were able to fully regenerate their tendon, in contrast to adult mice where only scar-dominant healing occurred. And this made us consider a potential role of neonatal progenitor cells, or TPCs, as biological augmentation method. In total, 52 adult mice underwent an acute supraspinatus detachment and repair and were randomly allocated to receive either a fibrin-only gel bead or a fibrin bead with TPCs incorporated. Outcome analysis consisted of cell analysis by immunofluorescence, functional analysis, and structural analysis. We hypothesized that delivered TPCs would participate in tissue repair and improve functional and structural healing. Now, prior to our main investigation, we looked at ways to try and label our applied cells, and we decided on using an adenocree virus, which utilizes Rosatidy tomato reporter of our applied TPCs to be able to track and locate our cells after implantation. For our quantifications, we used 2 different regions of interest, one being the neo-enthesis, and the second one being a tendon scar region, which were both morphologically based on our structural stains and then transferred to our relative antibody overlay images. Our first immunofluorescence outcome analysis was fibrocartilage by SOX9 staining, and we saw that TPC application increased the early enthesis fibrocartilage expression at the neo-enthesis region of interest. For scar tissue by laminin staining, which is a marker for extracellular matrix, we found that TPC treatment significantly reduced the amount of scar matrix formation across all our time points in both regions of interest. Similar diminished values were found for alpha SMA staining, which is a scar marker for myofibroblast cells. However, here there was only a significant difference at day 7 post-op at the neo-enthesis region. Our first functional analysis was Gait, which is an excellent surrogate measurement for shoulder function. And we looked at paw area, stride length, and stance time. In our fibrin-only control group, we saw that stride length and stance time did not recover by day 28 post-op, indicating that the application of our fibrin-only bead in combination with our detachment and repair model does not restore functional gait. This was in contrast to our TPC augmented group, where stride length and stance time did recover by day 7 and by day 14, respectively. Our micro-CT analysis showed that the cortical thickness of our TPC-treated group remained unaltered in comparison to non-surgical controls, and this again was in contrast to our fibrin-only control group, where a significant difference was found, suggesting that TPC treatment leads to an altered bony response after injury and repair. For our biomechanical testing, however, we were not able to show any significant differences between the 2 groups while testing the tendon-to-bone attachment. And in addition, we found that both structural and material properties remained significantly impaired after our model. For structural analysis, we looked at the enthesis structure and at collagen orientation. And by using toluidine blue staining, we were unable to see the distinct enthesis structure or regeneration in any of our specimens. However, we were able to show superior collagen organization at the enthesis in our TPC augmented group. Now, in conclusion, the results of our study show that TPC application restored and improved gait and altered the bony response relative to our fibrin-only control group. This may be due to the significant reduction in scar matrix formation, improved early enthesis fibrocartilage formation, or enhanced enthesis collagen orientation. However, we were not able to show any structural regeneration of the distinct structures of the enthesis, and there was no direct engraftment of our applied cells at the repair site, which we were able to evaluate by using the Rosati expression overlay images. The beneficial effects of our TPC application are therefore most likely due to paracrine signaling rather than a direct contribution of the cells to the repair. Future work will try and determine factors secreted by the TPCs and elucidate mechanisms to develop further translational targets. Our results just got recently published in JSCS, and I would like to thank and acknowledge all lab members that collaborated with this project, as well as Dr. Leesa Galatz and Olivier Verborgt. I would also like to thank the funding institutes and especially SECEC for the basic science grant, which made this project possible. Thank you for your time.